Hi, everyone. Welcome to Foresight's Biotech and Health Extension Group, sponsored by Handels Plus Capital. I'm super excited to have Justin Sonnenburg here. And before we hit the road running, I do want to remind people that if you're interested in joining us at Vision Weekend, it's coming up in San Francisco on December 2 and 4, and in Chateau de Fay in a castle in France on November 18 to 20. I'll drop the link here to that weekend in the chat. But it's basically an opportunity for all of you guys to meet people in this group and in other groups. We have a really exciting longevity and biotech track, but then we also have a molecular machines, neurotech, space, computing, and other tracks. And so if you are interested in just exploring a bit what progress could look like across different domains, I think that's a good one to go to. There's an application form. So that means that people here in this group, who are researchers, scientists, and so forth, can just apply for subsidized tickets. And then we'll get to that. And so we're in the final review of applications. So just a reminder, if you want to join, now is a great time to apply. I also want to say the Biotech and Longevity Report is now out in the open. Plus, so are our workshop videos. So if you want to take a look, please do so too. And there's also the longevity prize that you can still apply to, which I'll share a little bit more about in the chat. So that's all from me. For now, I'm extremely excited to welcome Justin Sonnenburg. We have taken all of your respective applications and nominations very seriously to give talks, which I'll also share again here in the chat. But this is just to say that, we're relying more as this group is growing on nominations from all of you guys to nominate wonderful folks that should be giving a talk. And I think, Justin, you have not only been nominated by one, but like several different people. So congratulations on that. And so we took that seriously. And here you are. Thank you so much for joining to discuss the gut microbiome. I will be in the chat. I'll be monitoring after your presentation. We'll get to the Q&A. As always, time gets very short at the end. So if you do want to make sure that your question gets heard, drop it in the chat so that we know that, we get to that early on. And for now, I'm really excited to have you here. Thanks a ton for coming. Thanks everyone for joining. And I'm really excited for the presentation. Thanks.
Okay, awesome. Thanks so much for that great introduction. Let me make sure I share the right, this is it. Okay, wonderful to be here. Yeah, thank you for the nomination and the invitation. So I wanted to today, cover the basics of the gut microbiome, but also what I see as a major opportunity. I think a lot of people are thinking about the gut microbiome as this interesting component of our biology and health, and thinking about it in a very traditional way in terms of how it can be leveraged in biomedicine. I think it's a little bit flawed, or at least not comprehensive. And I think the major opportunity is actually thinking about the microbiome in a completely new way. I don't know. I I think I see there's an admit that comes up. I don't know if anyone else.
That's the thing, as a co-host, you and I see it when people are trying to join, and I got that. So don't get distracted by people wanting to join. We're in high demand here.
I wasn't sure if I was the only gatekeeper, so wonderful.
No, I'm here.
Okay. So I think there's a major opportunity in potentially correcting gut microbiome deterioration in the industrialized world, and that could reverse trends of a lot of Western diseases. And I'll get into that in more detail. So I like to start off with this cross-section of the gut microbiome in situ. This is just a cross-section of colon. The microbes living in the lumen of the gut are in the top left here. Every single little colorful rod there is a different bacterial cell. And then the bottom right, the blue area, is host tissue. Those are colonocytes. And so you can see how close the microbes are to host tissue, only separated by this thin green mucus layer that we secrete to keep microbes in the right spot. So an incredible interface for exchange of biological information. We're still really we need to do a lot of work to map all of this, but we've learned some great principles over the past 10 to 15 years in studying this community. So we know that our human cells are outnumbered by the microbial cells that live in and on our body. So by cell number, we're actually more microbial than we are human. There's a vast coding potential in the composite genome of our microbiome. Most of these genes, we still don't understand what they do. And there, of course, is a major move to understand not just what microbes are in our gut, but to understand what these microbes are doing, how they integrate with our biology. We've known for a long time our gut microbes are important for gut health, important in helping us digest food, but there really is a major role of the gut microbiome in tuning a lot of our biology, ranging from metabolism to immune function to things like neurodegeneration, moods, behavior. And so this little diagram here kind of uh, captures the microbes as being upstream or in control of a lot of our biology. But I think a better way to think about it is just our microbes are part of the integrated system that is our body. What's special about our gut microbiome is that it's malleable, so we can change it, and it also is individualized, and so that gives us a tremendous opportunity for um, once we understand how to manipulate this community and how those changes will ripple along our biology, a tremendous lever for changing our health in various scenarios. And I really believe that the microbiome is going to be an incredibly important part of precision health going forward. But I think the same feature of malleability also leads to a vulnerability that our microbiome can deteriorate. And in fact, since we haven't really paid attention to it over the past several decades, over the course of tremendous lifestyle changes in terms of processed foods, antibiotic use, a lot of sanitation measures that during industrialization, and particularly over recent industrialization, that our gut microbiome has changed pretty drastically, and what are the implications for human health? So a major question has become, to what extent can we manipulate our own biology via the microbiota? Can we generate a knowledge base and set of tools to rationally manipulate these microbes? And there's a lot of different approaches for doing this, but one of the major things that you'll see if you look into the field, and certainly throughout the course of my talk, is that diet is the major tool that we have for manipulating our gut microbes in terms of composition and function. And so what components of diet are important? Well, it's actually dietary fiber. And I think that, dietary fiber has become known as kind of this boring, inert substance in our diet, but it really plays an incredible role in fueling our gut microbiota, and particularly what we like to refer to as microbiota accessible carbohydrates, the complex carbohydrates that are metabolically accessible to our gut microbiota. And this schematic just sets up the logic for why these components of our diet are disproportionately important to gut microbes. If we're eating a Western diet full of fat, starch, protein, simple sugar, a lot of that gets digested in our small intestine, absorbed into our bloodstream. And our microbes then are left starving. If we're eating a plant-based diet that's high in dietary fiber, we actually can't digest these complex carbohydrates. These end up making their way to the distal gut, and our microbes then can ferment these complex carbohydrates, producing short chain fatty acids and other metabolites that enter our circulation. And so there's really a huge metabolic difference between just eating a Western diet and eating a plant-based high fiber diet. If we're not fueling our microbes with dietary fiber, we know our microbes start eating us. These microbes can use that green mucus layer as a backup food source. It's also made of carbohydrate. And we know that a low fiber diet results in the bloom of mucin utilizing microbes, and also a thinning of the mucus layer in mouse models that we and others have tested. And so, one of the major shifts in our thinking came over the past 10 years in my lab, just thinking about the asymmetric plasticity of the microbiota over evolutionary time compared to our human genome. So we have these two components of our biology, microbial and human, that need to work together for us to function properly, but they can adapt at very different rates. So if you take antibiotics, you eat a Western diet, over a very short period of time, you can change your microbiome, whereas your human genome, of course, either as a person or as a society over generations, stays relatively static. And so this leads to the possibility that there could be an incompatibility between our human genome and our microbiome. And so we're really interested in mapping common features of the microbiome as humans spread around the planet. Can we learn about functions in species that may have shaped our human genome over the past 100,000 years and perhaps longer. And has the microbiome changed recently with lifestyle? And so we spent a lot of time thinking about these questions. We've been studying non-industrialized populations in different parts of the world. This was a study we published in 2017 focused on the Hadza hunter-gatherers, but bringing their microbiome data together with a lot of other microbiome data that have been collected in other studies. Our data is just in the line here that says Tanzania. Um, all of these populations, their microbiome data is represented on this principal coordinate plot that just separates microbiome composition based on variability in that composition versus other populations. And so basically it's the grouping of the populations on this x-axis that's meaningful here. And you can see the non-industrialized populations are on one side, industrialized populations are on the other side of the axis, suggesting or illustrating that there's been a change with lifestyle. This separation is not just general loss of biodiversity in the gut, it's actually loss of specific families of microbes. So these plots at the bottom show fractional representation of different groups of bacteria that live in the gut. And how their fractional population is represented across the same axis. Spirochetes, Succinate Vibrio, and Prevotella, all very abundant in non-industrialized populations, but rare or absent in industrialized populations. And the opposite is true of Verruca microbia, blooms in the industrialized populations, but very rare in non-industrialized populations. And it just so happens that Verruca microbia are mucin-loving bacteria. And so the same data that we see in mouse models with a bloom of mucin-loving bacteria within the gut on a low-fiber diet, we can see across hundreds of individuals in populations that eat low-fiber diet. One of the detractors of this data is that all the data came from different studies, people with different human genetics living in different geographies. And so Ashish Jha, a former postdoc in my lab that now is faculty at NYU Abu Dhabi, went back to his native Nepal and surveyed 4 populations that live on a lifestyle gradient, but they have the very similar human genetics and live in a similar geography. He quantified differences in their lifestyle using a correspondence analysis of a extensive survey questionnaire, confirming what basically was known about these populations, that the Chipangs are foragers, the Raji and Raute are transitional populations settled in the 1980s. They forage a little bit, but mostly farm, and the Taru only farm. So they settled 250 years ago. And so Ashish was interested in how their microbiomes compared to the microbiomes of Americans. And we see that the microbiome composition on the bar chart on the left, the composition of it mirrors lifestyle. So foragers are on the far left, Americans on the far right, and then the transitional communities are in between. So the extent to which populations have departed from a foraging lifestyle determines the extent to which their microbiome has changed. You can see that in more detail if you look at the different taxa abundance across these different populations. These are the taxa that explain separation in the data. So the individuals in the Chipang group are shown on the left here. Each column is a different person's microbiome. If we just compare the Chipang with the Americans, we see actually very similar differences to what we saw when we looked at the Hadza versus Americans. But now with these transitional communities, we can actually capture intermediate states of microbiome transition. Again, the extent to which populations have departed from foraging determines the extent to which their microbiome has changed. You see very similar patterns, Akkermansia mucinifila, very low abundance in the traditional communities, very abundant in the Americans. So this is a mucin-loving bacteria. The opposite is true for these species that we term vanish taxa. So these are species or families that are volatile and associated negatively with industrialized societies of humans. So these are the microbes that are disappearing in the industrialized gut. And so Ashish, in this PLOS paper that we published, proposed this model of disappearance and appearance of different microbes across this lifestyle gradient. I mentioned the vanish taxa. We also note the blossom taxa. These are taxa that bloom or selected in societies of urbanization and modernization. And Ashish noted these different patterns of disappearance and appearance. And we think about these populations as modern populations living different lifestyles, all equally evolved. But another way to interpret this data is the change in human lifestyle over time for industrialized populations. So we all were foraging at one point about 10,000 years ago, switched to agriculture, and now over the past 1 to 200 years have become increasingly industrialized. And now our lifestyle is spreading around the world, changing the microbiome as it spreads. So that's all compositional data. What about functions? We have metagenomic data now that confirms a lot of these trends of composition, and then gives us great detail in terms of function. This plot just shows functions that are enriched in the hunter-gatherers versus enriched in the industrialized gut. There are major differences, and so this tells us that these changes in composition are not just changes in that result in redundant functionality with different species performing the same functions. We actually have a functionally different gut than we had probably just a few hundred years ago. And again, with the same human genome. What's striking about this data is the number of oxidative stress genes that are represented in the industrialized gut. And this is a pattern that suggests there's either simmering inflammation or at least a different metabolic state within our epithelium and colonic tissue. So really interesting trend that is consistent with or supportive of the spread of things like inflammatory bowel disease in the Western world. And so this change in the gut microbiome can actually be observed in real time. This is a beautiful study from Dan Knight's group published a few years ago, where they followed immigrants from Southeast Asia to the United States. And what they saw is that within months, the microbiome of these people would change to look more American. The longer people were in the United States, the more diversity they would lose. They would lose Prevotella species, which is a hallmark of industrialization. They would lose fiber degrading bacteria. So again, a low fiber diet, you lose a species that degrade fiber, more Bacteroides strains. And then the pattern becomes more severe over generations. So US born second generation children of immigrants have yet a more industrialized gut microbiome. Again, these are all, modern populations happening in real time. Beautiful study out of Harvard from Alex Kostik's group sequenced paleofeces, North American individuals that lived 1,000 to 2,000 years ago. They determined the composition of the microbiome based on that sequencing data, and then overlaid the paleofeces in the orange spots here on top of other modern day microbiome compositional data from populations that are industrialized and non-industrialized. We see the paleofeces are very similar to non-industrialized microbiomes in terms of the microbial composition, supporting that the industrialized microbiome is a recently derived state, that the non-industrialized populations have microbiomes that are similar to the microbiomes of our ancestors. So we've changed our microbiome recently. Our microbial identity has changed. We know our gut microbes can change our immune status. We also know that chronic inflammatory diseases are on the rise, whether you're looking at ulcerative colitis, other autoimmune diseases, food allergies. There's just a whole host of immune dysregulation driven diseases that are both have increasing rates and earlier age of onset and spread as our lifestyle spreads. So the question is, could this foreign microbiome inside our guts be dysregulating our immune system? And, we have an approach that we've kind of inherited from the era of infectious disease of reactive medicine, of waiting for chronic diseases to arise, and then basically trying to treat the symptoms of this inflammation by shutting down the immune system, which has a lot of negative consequences. But, tons of drugs try to shut down the immune system or aspects of it in inflammatory disease. So it's like we're trimming the branches of these diseases as they arise. And instead, a major question is, can we think about this differently? The current approach I liken to this cartoon. The sailor says, "Great news, Captain. You can inform the passengers we have slowed the rate of sinking." And instead of kind of slowing the rise of diseases, I think the question is, can we stop them in their tracks? What are the root causes of these trends? Are there we have dozens of these inflammatory diseases. Are there really dozens of different causes, or could there be just a small handful of causes? And then what disease you develop is determined by either environmental factors or maybe elements in your human genome that determine that you'll get heart disease and I'll get, cancer. But basically it's inflammation that's really sending us in these, on this trajectory. So, could the microbiome and diet really be at the heart of this? Is there a unifying theory? And so we really started thinking about, can we repair our gut microbiota and improve our health, or at least test if health can be improved via microbiota change? And we started the Center for Human Microbiome Studies at Stanford with a great group of partners. And the goal is really to develop humans as an experimental model for studying the gut microbiome. Um, we, I think, I work closely with my wife, Erica. We were raised kind of in the era of thinking about molecular mechanism, working up through animal models, and then trying to make a translational leap to humans that typically fails. And I think probably has a higher failure rate when it comes to the microbiota, just because of the divergence from animal models to humans. Because we're studying diet, because diet's safe, we thought, well, why can't we start just doing experiments in humans with diet, use omics data to map interactions and then reverse translate back into animal models, knowing that we'd be studying something that's relevant to human biology, and also get information right up front about what does and doesn't work for changing aspects of human biology via the microbiome nutrition axis. So we've performed a bunch of studies in close collaboration with Christopher Gardner's group. I wanted to just provide one example of this. This is a study that we call the E5FO study, where we fed people either fermented or high fiber food over the course of several weeks to see how that would impact their microbiome and immune status. We published this in Cell last year. It was covered in the New York Times if you want a quick read of the study overview. But basically, I'll just give you a little bit of the highlights here, focusing on the group that ate high fermented foods. So these are people eating, kimchi, yogurt, kefir, sauerkraut, kombucha, things like that, foods with living microbes that have been fermented. We got people to eat differently with the help of Dahlia Perlman, who's an incredible diet educator at Stanford that works with people to get them to actually get free living humans to eat differently, which is not so easy. And then the project was really propelled by three fantastic trainees, Maddie, Hannah, and Gabby. And Gabby's now faculty up at UCSF. So this is a quick overview of the data. We got people to increase their fermented foods from baseline up through the maintenance phase. So here we enroll people for 3 weeks. We monitor blood and stool throughout the course of the study. We take them through a ramp phase, a maintenance phase for 6 weeks, and then a washout phase 4 weeks afterwards. During the maintenance phase, we got people up to over 6 servings of fermented foods a day. They started off at about 0 servings of fermented foods a day, so a big increase. We saw that over the course of this increase, big increase in their microbiota diversity. This looks messy, but in the context of human studies, this is really a notable increase in diversity that frankly is not commonly seen with dietary interventions in people over this sort of time frame.
Question on that slide, question on that box plot.
Yeah, please.
So that does look quite messy. And I'm curious, is the linear fit least squares to the medians, or what are we looking at there?
Yeah, I don't remember what the fit was. I can send the paper and make that available.
Great.
The data that I'm not including here is the high fiber data, and then a lot of other human study data where the line is just completely flat despite the intervention. That happens over and over and over again. And so, so we do see significance from baseline in several of these later time points.
Okay, follow-up quick question. Yeah. So the whole fiber thing is kind of really dubious, right? In my opinion, at anyway, my limited look. And I'm wondering, are you separating out, is fiber, like this says fermented fiber food, like there's kind of two things there. There's fermented stuff and then there's fiber. They're separate issues, are they not?
Totally. And what I'm not showing here is the high fiber arm. So we had another arm that ate legumes, whole grains, vegetables, fruits, increased dietary fiber from 20 grams at baseline up to over 40 grams of dietary fiber for 6 weeks during the maintenance phase. So I'm not showing that data for the sake of time. But interestingly, in that study, in that arm of the study, there was no increase in microbiome diversity. And I'll show you in a second here, for the fermented food arm, there were major changes in, I'm just showing 4 here, there were over 24 inflammatory markers that decreased as these people were increasing their microbiome diversity consuming fermented foods. We saw no changes like that with the dietary fiber. So we had 2 separate arms. I'm just showing the high fermented food here.
And that was 6 weeks of duration?
Well, so these plots show over the time course. So it decreases as the study progresses. But the major endpoints of the study are from the what we call 10 weeks, but it's 6 weeks of maintenance on top of a 4-week ramp period to let people's guts equilibrate to the diet. So we're comparing the endpoint to the baseline. So the microbiota diversity is definitely crashed in the industrialized gut. It's probably happened progressively over a long period of time as we've changed our lifestyle. We can talk more about the dietary fiber intervention and why that failed over that short period of time. I think this is probably likely due to the fact that we don't have the right fiber degrading microbes. As that study on the immigrants showed, you lose fiber degrading microbes in the industrialized microbiome. And if you just bring back dietary fiber, there's a good chance that it would just shoot right through you unfermented. And it's really the short chain fatty acids that are produced when fermenting the fiber that are the immune modulators. And so it's not surprising that in industrialized guts over a short period of time without providing fiber degrading microbes, we're not seeing a significant change. We may need to reintroduce fiber degrading species to actually see an effect over a short period of time.
Is it possible that 10 weeks just wasn't long enough for small numbers of those species to evolve to the larger numbers?
Yeah, exactly. I think when you combine a short period of time for an intervention and a sanitized lifestyle where we don't have a lot of access to gaining microbes over a short period of time, that you're just kind of stuck with this industrialized gut. We did monitor carbohydrates in stool and saw a lot of what should be fermentable hemicellulose carbohydrates in this stool, suggesting that indeed over this short time period, you just see these fibers zipping through the people. I think if you were to implement a high fiber diet over months and years, you would gradually acquire those high fiber microbes. And actually, I was just going to spend a few slides talking about synthetic biology.
I'll just note before you move on to the next topic, but you said 20 grams to 40 grams in that high fiber intervention.
Right.
And, if you eat only unrefined, unprocessed whole plant foods, that diet provides 60 to 80 grams of fiber per day. So even 40 is a sort of low number compared to what's achievable.
I totally agree. And if you go to the Hadza hunter-gatherers, they eat over 100 grams of dietary fiber per day. So, I think the 40 grams was above the recommended guidelines for most diets. It's based on the amount of dietary fiber per calories that you consume. But usually, I think even for, 2,500 calories, it's less than 40 grams per day. Most of that's based on heart health data. And I think probably more fiber is likely healthier in more in most cases. So anyway, I agree with your comment. But the second part about synthetic biology actually sheds light on the idea of acquiring microbes that can degrade fiber, although it's more focused on the technology of using that to engraft microbes into the gut microbiome to perform specific tasks. So I've mentioned the gut microbiome is a important component of our biology, but we can also think of it as a technology platform. These microbes are rich in sensors. They can detect all the nuances in the gut environment because their survival requires it. If we can learn about those sensors and rewire them, we can actually have microbes that respond to specific conditions in desired ways. And so we've been working on this just to, probably most of you are familiar with synthetic biology, but the idea is just to generate a parts list that you can wire together genetically that gives you a desired predictable outcome. And so we focused on the Bacteroides, the most abundant genus in the American gut, as a way to, as a chassis for engineering the gut microbiome. And I should say it was on the initial slide. The two scientists, Wes and Liz, who were on that first slide, have gone off and founded this company, Novome Biotechnologies, that I'm a co-founder of, and are now in phase 2 trials. And I'll tell you more about the technology that they're developing. So, synthetic biology allows you to do things like make gut bacteria that express different fluorescent proteins. Here we're looking at 6 different species of Bacteroides within a mouse gut. Here living within plant cell wall. So that's the purple area here. You can see the different species have different colors, different kind of clonal populations, but also well-mixed populations, using the cell as actually a little room and potentially degrading fibers off of the cell wall there. So one of the major barriers to using microbes in the gut as a therapeutic is the barriers to engraftment. It's been well studied, well described that if you put a gut microbe into several different people and track it, it will do very different things in those different people. And this was nicely shown with the Bifidobacterium longum species that was fed to people for 14 days. This just shows the density of that B. longum in these different individuals. So each line is a different individual's B. longum density over time. After cessation of the treatment, about 2/3 of the people cleared the B. longum, but about 1/3 of the people kept B. longum within their gut, shedding it. These are known as persisters, where B. longum engrafted. It turns out that the people that engrafted B. longum lacked a similar strain at baseline. B. longum was lost from individuals that had a similar strain of B. longum at baseline. So it is consistent with the idea that if you possess a strain in your gut, the second arriver of that same strain will be lost, will not have a place to engraft in the gut. So this is a big problem if you want to develop a microbial therapeutic. And so Wes and Liz took this on. They first wanted to demonstrate that variability, where they took a surrogate therapeutic microbe that we could track, put it into 3 different groups of mice, one harboring a mouse microbiota, the other 2 harboring 2 different human microbiotas, just track the density of that microbe over the first week. You can see very different dynamics, high levels and dwindling, mid levels and dwindling to below the level of detection. So very different dynamics depending upon the background microbiota that this microbe encounters. We actually isolated this microbe for its ability to degrade a very specific polysaccharide from seaweed known as porphyrin. So this seaweed polysaccharide, because it's not commonly consumed by Westerners or by mice, there's no background competition. And when you provide seaweed in the diet of these mice, you actually are providing a privileged niche specifically for this microbe without any background competition. And so at day 7, we started feeding the mice the seaweed. And here we see what happens to the density of the microbe. In all 3 cases, in all 3 groups of mice, we achieve high and reproducible levels of density during that seaweed extract feeding. And what's more impressive was Liz decided to titrate the porphyrin in different groups of mice by tenfold dilution. So 1%, 0.1%, and 0.01% porphyrin. We see that the density achieved during that feeding shows a direct linear relationship with that porphyrin dose. So log decreases in the density achieved, suggesting that you can actually tune the abundance of this single microbe in the background of a complex microbiota. And that's shown in images here, where our microbe is labeled in green and the background microbiota in pink at both low dose and high dose porphyrin. So you can actually modulate the abundance of a single microbe within the gut using this privileged niche strategy. And so Novome has taken this into human trials now. This is human data that they recently released, again, using a range of doses of porphyrin, giving the therapeutic microbe. With no porphyrin, they don't see engraftment, and then they see engraftment at the level that corresponds to the dose. And when they withdraw the porphyrin, the microbe, the therapeutic microbe is lost from these individuals. So this is now a system where you can engineer in other systems for desired functionality in the gut microbiome. And this is what they're using as a therapeutic chassis. And it makes available a lot of possibilities for gut targeted therapeutics. And so just to finish up, thanks to my group, wonderful collaborators, funders. My wife and I have written a book if you're interested in learning more about the microbiome, and look forward to the discussion.
Wonderful. Thank you so much. Okay, this is quite something. I think Carl has already applauded you as the very first one up in this chat for actually making the book available as an audiobook as well. You're getting lots of applause here. Okay, let's jump into the questions. I know that we had a few of them already clocked pretty early. This is also now to say, if you do want to just raise your hand and not drop the entire question in the chat, you can do that too, and I'll get to you as well. But I think perhaps starting with, I think it was first all the way up Steve's question, or Cosmo. Cosmo was even before that. So maybe Cosmo, you go for it. If you can unmute, otherwise I can ask the question.
Were you asking me or were you asking Cosmo?
Yeah, Cosmo, you go first, and then Steve.
Cosmo has no mic.
Well, if
All right.
Mike, he should text it.
Yeah. Any thoughts on why Akkermansia monocyphilia are beneficial to health, especially given their preference for feeding on mucosa?
Yeah, it's a great question. And I think it's one of the big capers. It may have to do with dosage. It may have to do with difference in strains. It may have to do with the fact that a lot of what people suffer from, and where Akkermansia seems to show an effect, is in that I think the Akkermansia is showing positive effects in metabolic syndrome, perhaps enforcing the gut barrier, and in potentially stimulating the immune system in immunotherapy for cancer with checkpoint blockade. In those cases, it may be that Akkermansia is actually goosing the immune system to do things that maybe are not in our best interest for longevity, but ameliorate. It's kind of like the keto diet does great things for. And I don't want to get into a whole keto diet debate here, but my opinion of the keto diet is that it can ameliorate a lot of metabolic dysfunction and problems with our high carb diet, but it's probably not healthy long term. And so my feeling for Akkermansia is kind of along those same lines.
Wonderful. We have Steve next.
I'm really interested in the clinical issues surrounding this kind of potential therapy. And I'd like to know if how the technology is developing or the science is developing around the questions of looking at microbiome composition pathologies as different from, let's say, mucus layer deterioration or the failure of tight junction gaps from, let's say, inflammatory induced zinc deficiency.
Yeah, it's great questions. And I think one of the big issues that the field is experiencing is all of the feedback loops that are present, where you end up with microbiota that reinforces physiologies that may not have started due to the microbiota. So I think a good example of this is, the OB mice that we know lack a satiety peptide, just eat a lot. They end up obese with a different microbiota. If you transfer that microbiota to lean wild type mice, those mice will actually gain weight more rapidly than mice that receive a normal microbiota. We know that microbiota did not initiate obesity in the original case, that it was the genetic deficiency of the satiety peptide and just eating a lot that did. But you end up with this microbiota that potentiates that metabolic feature. And so I think in the case of like barrier disruption and, altered microbiota, you can go into these diseases, see altered microbiota. That microbiota in many cases probably reinforces and potentiates disease. Whether it is causal in terms of initiating, I think remains a major question. But my guess is actually let me flip to a slide here that just may clarify this a bit. I frame this in terms of dietary max, short chain fatty acids, and MAC consuming microbes. But I think that this kind of captures, an industrialized microbiota that doesn't produce a lot of short chain fatty acids. We know short chain fatty acids reinforce the barrier. This microbiota consumes the mucus. You end up with this eroded mucus layer, all kinds of inflammation occurring. The host reacts with reactive oxygen species, increased oxygen tension in the gut. You end up with more facultative anaerobes here, a less anaerobic microbiota that then potentiates a cycle. And the opposite is true for a healthy microbiota where you can produce short chain fatty acids. You have a healthy mucus layer and you maintain low oxygen in the gut. So I think that you can end up in these cycles and then unraveling kind of where to intervene or how many nodes you'd need to intervene at to have an effect is a major question for the field right now.
Okay, I'm collecting questions as we go, and we have a ton. So let's do a rapid question fire round here. I'm sure that we add more as we go. Next question we have from Micah. Micah, if you have a mic, ask yourself. Otherwise, I'll hold it out. Awesome.
So if we can sequence the microbiota of paleo fecal matter, can we then synthesize that same basic cocktail and then use that as a transplant cocktail basically and give it to people and on a recurring basis to try to get people back to what we think is at least what we were evolved with? And if so, do you think that would be useful?
Yeah, I think it's a great question. I think that we've probably lost species and functions that are important for our health. Whether all of them are is debatable, probably not. And so to figure out which ones we should and should not bring back, what features of our current microbiome are adaptive for our current environment versus those that are detrimental are major questions. I will note that, there are instances of people doing fecal transplants on themselves from the Hadza hunter-gatherers, which is similar to the experiment you're suggesting. You can read blog posts about that. But the one item just to quickly note about Novome here, they actually have engineered this microbe to degrade dietary oxalate to prevent kidney stones. Because if you get kidney stones, you're likely to recur. Most kidney stones are made of calcium oxalate. And the doctors will just tell you to avoid oxalate-containing foods. But it turns out that if you look at we used to have a microbe in our gut called Oxalobacter formigenes that could degrade oxalates. That's one of these vanishing taxa. People that have it are protected from kidney stone development, and yet we know it'll disappear over time, most likely. And so Novome is bringing this back, not because they're interested in rewilding the gut microbiome, but because they're interested in a therapeutic, but essentially they're conducting a functional complementation to a major function that we've lost. So you can do this function by function and maybe get to this answer, but it's a good question.
Do any of the microbiome testing companies test that specific one that we've lost to see, which individuals, so, could individuals tell whether or not they have it or not?
Yeah, I don't know, and I don't know the level that it needs to be at for it to be functional. So it's a good question, and I actually don't keep up with what the testing companies are doing because there's a lot of them, and data quality varies from what I've heard. So anyway, buyer beware. But if you see Oxalobacter formigenes on your report, that means that you have this microbe that can degrade dietary oxalate.
Cool. Giacomo next.
Hi, do you hear me?
Yeah.
Yeah. Actually, you partially answered my question a bit, but I was curious about generally what are the kinds of therapeutic uses of the artificial bacteria that you were talking about, like Novome is developing. I mean, you just gave an example right now, but I was curious about like kind of the range of things you could tackle with this.
Yeah, no, there's really good examples. You can go back. There's a science paper in 2000 where people engineered Lactococcus lactis to secrete interleukin-10 to treat inflammatory bowel disease, and that went into the clinic. And I think it was a dosage issue. It ended up failing, but there are examples of delivering cytokines, and you could imagine maybe incretins for metabolic disease. So I think there's a large range of possibilities for delivery of therapeutics from the gut. And, of course, GPCRs that are being mapped right now where you could imagine microbes secreting metabolites that would trigger them as well.
Cool. Wonderful. Next one up, we have Johnny.
Yeah, you partially touched on this as well, but I was wondering what other experience you've had with analyzing the microbiome for an individual person, then coming up with a custom probiotic mix. And in the chat, I entered my experience with the Flore Sun Genomics mix. I was wondering if you had any thoughts on that. They first analyze your microbiome, then create a custom pill that you take every morning. And then in a couple months, you reanalyze it. So
Yeah.
I'm about to get mine reanalyzed. I'll probably send off a sample.
Yeah, great.
As well.
Yeah.
So go ahead.
My opinion is that's all crap. And I don't mean literal crap. I mean figurative crap. I think the ability to personalize probiotics to the gut microbiome is totally premature and half baked. I don't know that company except for things that I've heard, all hearsay. It supports my theory. I will say that the real opportunity here, from the standpoint of impact, not making money, is to think about population level interventions that can, because that what has happened with our microbiome, with this deterioration, what's happening in industrialized populations as our lifestyle spreads, is affecting hundreds of millions of people, to the point where the Gates Foundation is becoming more interested in metabolic disease than infectious disease over time, because this is afflicting all of these nations that are starting to eat processed food and gain access to antibiotics. And so if you want to have an impact in this field, you should think about not personalized, but population level interventions that can fix the microbiome for hundreds of millions of people.
Well, thanks. And I'll probably wind up going back to the fermented foods and other dietary. Thank you.
Yep.
Wonderful. Near, you're next.
Terrific, and thank you. I've enjoyed your science, but I'm enjoying your wisdom really a lot. So I'm going to make two statements to provoke you. Tell me if I'm wrong. We're interested in aging, and as far as I read the literature, microbiome doesn't change with age, not cross-sectionally, and not individually. Although I wouldn't say that we have from 0 to 100 right yet, but and even if you give antibiotics, it returns to its original within a year or probably shorter. So we're interested in aging. I just wanted to know if you picked some. That doesn't mean that the microbiome is good in individual, but do you think it changes with aging? And second, a drug, a gerotherapeutic like metformin, there are several studies that says that it changed the microbiome and even suggested it's a mechanism of action. You know, do you think of drugs as changing microbiomes?
Yeah, super interesting. So the first point, I think it's very clear that the microbiome changes really drastically over the first 2 years of life. So, we're born without a microbiome, and it's like an ocean, an island rising up out of the ocean. This new ecosystem, our gut, is rapidly colonized. There's definitely a succession pattern. And actually, we recently, just this year, published a science paper looking at infant microbiome development in non-industrialized versus industrialized populations. And that pattern of succession of microbiome assembly is very different in industrialized versus non-industrialized populations, with a notable microbe, Bifidobacterium infantis, that's the canonical degrader of human milk oligosaccharides, being lost in industrialized populations, even in infants that are exclusively breastfed. So this is like a population-wide loss of a very important gut microbe for infants. So that part of life, there's definitely change. I agree that over the course of the bulk of life, it's very stable, resilient to perturbation or resistant, even in some cases. And then, toward the end of life, there can be different trajectories. There can be, loss of diversity. This is usually I think the model right now is that this is brought on by a change in eating habits as people progress, particularly in care facilities as they get older. And this corresponds to like immunosenescence and inflammaging and things like that. But I think that there is this question of like, the stability of the gut microbiome throughout life and what this means to an individual, and particularly thinking about disrupting that or reprogramming it. So that's a really good question. Your second question was
drug like metformin changing microbiome.
so I don't know a lot about metformin except for that it may act as an antioxidant or reductant in the gut. And this gets back to my model of that we are in a redox state that has too much oxidative stress for the gut microbiota to be healthy. And so it may be that metformin helps to return it to a less oxidative state, but it could be other modes of action. There's no doubt that drugs can have a massive impact on the gut microbiome. And this may be beneficial in some instances, it may be deleterious in other instances, but it may also provide a powerful lever for reprogramming the gut microbiome in various ways going forward. So yeah, I think they have a role to play for sure.
Thank you.
Wonderful. Carl.
So I have a sort of a general question, and then the specific instantiation is, that you showed several slides early comparing various populations. First on the ones, the 4 different populations from Nepal against modern sort of Western, and then later the sort of, real paleo, versus, newer, versus, anyway. So I would love to see, I wonder if data is available that adds extra, modern people, but eating super like plant-based, 80 grams of fiber a day kind of diets, whole food or typical whole food plant-based diet. Like I'd love to see where those fall relative to the US versus those 4 different Nepal people, and that might help answer that question of, how much of the modern changes are compensatory versus, problematic. Because the high level, so I'm curious if that data is easily available somewhere. And then the high level question, of course, is that, you're the expert here, but I know you're a researcher. And so I understand you're not following the sort of available now direct to consumer type testing companies and things like that. But, who do we go to help be the most expert opinion on what to do now? And I know you have some of this in your book. And of course, there's this more recent book called Fiber Fueled by another gastroenterologist who's got some expertise. And there's some good advice in those books. But
Yeah,
that's the high level question. Who does one go to for the optimal thing to do now while the research continues?
yeah. It's a good question. I think I love the idea of going to people that are eating a super healthy, high fiber diet for a long period of time and seeing if their microbiome is different, if their metabolic status and immune status.
And there are people doing this, right? I mean, there's a whole calorie restriction group, and people like Luigi Fontana have done other studies on them, but they haven't sequenced the microbiomes that I know of. People like Dean Pomerleau and myself, we have like 70 grams a day or more of fiber consumption, for example.
Yeah, totally. Yeah, no, I would love. One of the incredible limitations in this area is funding. I mean, all of the dietary intervention work. So actually that fiber fermented study we're now doing in pregnant mothers to see if we can change the microbiota that they pass to their babies and whether that changes immune status of the infants. We're proposing to also do this in kids with food allergies right now as well with fermented foods and fermented food derived products that we could translate into medical foods to infiltrate the clinic. But I think that the major challenge here is funding, because the National Institutes of Health has just like tiny allotment for nutrition studies. Very difficult. We've had many grants rejected, even with preliminary data of a cell paper. So I would love to do studies like this. If you have funding to do those sorts of studies or could target me to those people, it would be a study I'd be interested in doing. I think, in terms of where you should go for optimizing your gut microbiome, I think if you're dealing with something like metabolic syndrome or prediabetes or something like that, there are these companies like Date2 that are very solid scientifically, that are using machine learning based on microbiome profile to recommend what people should eat to keep blood glucose spikes low postprandially. But I think that in terms of the concept of maintaining health, and that's really like, I think where the money is here. If we can figure out how to get out in front of these diseases and tell people what they should be eating or microbes that they need to augment their microbiome with to prevent inflammatory disease, I think that that's really the direction that things need to head. And there's nobody that can do that right now.
Cool. We have 2 more minutes. I do want to get to the last question. And I know that you've already touched on this a little bit, but I just want to draw it out kind of like right in the open, which is the question of like, if people here in this group are excited about this work, which I think they are, then what is the best way that they can support this? Basically, like what are requests for collaboration, requests for funding? And I know that you've already like mentioned one part, but they may be more like, this is kind of the shameless plug moment. Yeah, give it to us. How can people help?
Yeah, fantastic. Thanks, Allison. I think that the major issue here comes back to funding. I think that, at Stanford with Christopher Gardner's group, we have a human immune monitoring center where we can monitor the immune system in high detail. So we know how people's inflammatory status changes. And then we have a variety of other technologies that we're using. So we have like all the components, technology, infrastructure in place. And what we're lacking is basically funding. And so, populations definitely help enrollment. If we can speed that up for various studies that we have funding for, that will decrease the cost of the study. But what we're really starved for is data, because, as Carl was mentioning, like people want to know what, should you be eating a Mediterranean diet? What are the different microbes that are important in that? What we need to do is generate a bunch of data where we can map diet to microbiome to different elements of health, including different components of the immune system. If we have that map, now we can start to tweak various microbes to get specific responses. So for instance, if you're going in for immunotherapy, you might want a very different immune status than if you're fighting an autoimmune disease. And to have a map of how to modulate the specific immune components based on a diet microbiome axis would be incredibly powerful. So I think that we need more studies, more data, and then we'll be able to take profiles and tell people where they're headed and what they need to do to fix it.
Wonderful. Thank you. That was loud and clear. I think we've really heard you. Thank you so much for joining us and presenting. I think people are extremely excited about your work. Thanks a lot. There's definitely a lot that's uncovered and a lot there's still to be uncovered. So really, really interesting. Thanks a ton for joining. For everyone here, I just shared in the chat the nomination form for future topics. So again, if you want to make your voice heard in nominating future topics, then that's the best way to do so. You can also always shoot me an email, which many of you have been doing. So thanks for all of that. Really, really appreciate it. Thank you so much for joining, Justin. I hope it wasn't the last time that we hear you here. And I hope you all have a wonderful time. Great rest of your day. And I'll see you very soon for the next seminar. Thanks, everyone.
Thanks very much. Take care.